Okay, thank you. First of all, I want to thank the organizers for inviting me to give this talk. Before starting, and in order to facilitate the understanding of the idea, I want to share with you, I think it's important to mention that the protein coding information represents only a portion of the total genetic load of a genome. In this case, RNA viruses, they have to carry all genetic information required for the successful completion of the viral cycles. And to achieve this, RNA viruses have developed different strategies. And one of them is to store information in RNA in discrete structural RNA units. Sorry, this doesn't. Okay. Thus, viral RNA genomes are composed of discrete RNA structural units that store important genetic information for the virus. They are universally conserved among different isolates of the same virus. These discrete structural units can be grouped in complex folded structural domains that are responsible of the essential functions of the virus. Altogether constitute the RNA genome. In such a way that more than preserving the sequence, what is important is to preserve the structure, the global structure of the RNA genome. And this structure is responsible of the function of the genome all throughout the different steps of the viral cycle. In this figure, I depicted the RNA genome of hepatitis C virus. But this is not a feature, this is a general feature for other viral RNA genomes. And in this figure, I have included, besides the hepatitis C virus, and two other viruses, like it's an example of a flavivirus and a picorna virus. And as you can see, all of them are characterized by the presence of all these discrete structural units that they are important, or they carry important information for the virus. They are universally conserved in structure, and this information they achieve, let's say, a role without the need of being translated into proteins. So the identification of these structural units and the functional characterization may provide a plethora of new potential antiviral targets. Well, in the first part of my talk, I am going to tell you about the work we have done in the lab characterizing the structure and function of the hepatitis C virus RNA genome. The genome of HCV is a single-stranded RNA molecule which contains a single open reading frame that is flanked by two untranslated regions. These untranslated regions are very rich and complex folded regions, rich in this or characterized by these specific or discrete structural units that they are universally conserved in structure. But this structural conservation is not restricted to the ends of the genome, and structural conservation can be found all throughout the open reading frame. So in this figure or in this representation, I indicate with these gray boxes the structural conservation all throughout the RNA genome. At the bottom, I have depicted few of these structural units that have been characterized. And I want to draw your attention in these three structural units, which are named 5-BSL-3.1, 5-BSL-3.2, and 3.3. They constitute the functional domain called cis replication element, or CRE, and that is coded at the 3' end of the open reading frame. So at the 5' of the genome, we can find an iris. The iris, which is formed by all these universally conserved structural units, is essential for translation of the virus. But the 5' has been also involved in replication. It's essential for replication. Similarly, at the 3' end of the genome, we can find a structural functional domain which can be folded into two alternative conformations, 3 stem loop conformation or a 2 stem loops conformation, in such a way that this conformation is competent for genomic dimerization. It exposes at the apical portion of this structural unit a universally conserved sequence, which is called dimer linkage sequence. It's a palindromic sequence which is responsible of the initiation of the dimerization. This domain has been involved in replication and also in translation. And as I had mentioned it, at the 3' end of the open reading frame, we can find this CRE functional domain, which is composed of 3 structural units, 5VSL 3.1, 3.2, and 3.3, which is essential for replication. So if both ends of the genome are involved in the same functions in replication and translation, it's easy to assume that both ends of the genome has to communicate each other. So the first question we asked was whether the RNA has any role in this communication between the two ends. So we did a very simple experiment in which we took 2 RNA fragments that corresponded to the 5' end and the 3' end of the genome. And as you can see in this figure, both RNAs in vitro, in the absence of any other factor, just the RNA, there is no proteins, they can come together and form a complex. Further, we demonstrated that this complex is absolutely dependent on the presence of the CRE element. And we know, we demonstrated that this complex depends on the interaction of the internal loop of this structural unit, 5VSL 3.2, within the CRE element, with the apical portion of this 3D structural unit that within the iris that is at the 5' of the genome. So they can come together and form this duplex. Further, we wanted to see whether this interaction that occurs in vitro in the absence of any protein can be functional. For this purpose, we made this construct in which we use the messenger of the luciferase gene flanked by the 2 ends of the hepatitis C virus genome. And we made a set of deletion mutants in which we removed the different structural units of the CRE element. And as you can see in this figure, just the deletion of the 5VSL 3.2 unit result in an enhancement of translation. So it means that this structural element is involved in the negative regulation of translation. And further, we could see that just modifications of the sequence at the internal loop of this structural unit result in the loss of this role in regulation of translation. So we detect again an increase in translation, whereas the modification in the apical portion, in the apical loop, it doesn't have any effect. So with these experiments, we demonstrated that this structural unit has the information to repress translation. So we demonstrate this interaction is involved in the regulation of translation. The work done by other groups demonstrated that this structural unit is also involved in this interaction that is required for replication. Another interaction with this other structural unit was also involved in replication, and for example, this other interaction has been related with translation and replication. So, as a summary of all this work, we propose that it exists a dynamic network of RNA-RNA interactions that is involved in the regulation of the essential functions of the virus. And in this dynamic network of interactions, it seems to be like that the 5VSL 3.2 structural unit plays a central role. Further, in order to know how the molecular mechanism, how these interactions can modify the function of the other functional domains, we did a structural analysis of the whole hepatitis C virus RNA genome. And we could demonstrate that the presence of the 3' end of the genome results or induce a modulation of the structure of the 5' end. So besides this interaction that we demonstrated between these regions or these sequences, the rest of the structural modifications cannot be explained by a direct complementary RNA sequences. Similarly, the presence of the 5' UTR modulates or modifies the structure at the 3' end of the genome. So basically, what we are these results are telling is that once again the structure of the genome is responsible of these changes in the functioning of the different domains. So in the regulation of the function of these elements. As you can see, that the 3' end, most of the structural changes induced by the 5' end accumulate in this 3' X-tail domain, which I told you that is involved in the dimerization of the genome. So we try to see whether these changes are involved in the change in the functioning of this 3' X-tail element. So in this case, in an experiment, just all in vitro, just with RNA, there is no other factor involved. We could see that whereas the 3' X-tail element can dimerize, when we add the CRE element, so this construct, it results in a 50% increase in dimerization. Whereas the presence of the 5' end of the genome results in an inhibition of this dimerization, which is even more evident when we remove the enhancer that is the CRE element. So basically, with this data, we can say that the efficiency of genomic dimerization is controlled by structural elements outside the 3' X-tail domain, and it confirms the existence of functional RNA-RNA interactions that involves the iris, at least the iris, the CRE, and the 3' UTR that modulate the structure, and this modulation of the structure modulates the functioning of the genome. Well, as I mentioned, this is not a specific feature of hepatitis C virus, and so we decided to study all close related viruses like they are the flaviviruses. The flavivirus genus includes different important human pathogens like yellow fever virus, dengue virus, West Nile virus, Zika virus, etc. This is a representation of the genome of the West Nile virus. This is a single-stranded RNA molecule. It contains a single open reading frame flanked by a 5' UTR and a 3' UTR. As you can see in this figure, the both ends are also very rich in these structural or discrete structural units that they are important or play important functions for the virus. In this case, it has been shown or identified complementary sequences at both ends of the genome that are involved in the interaction between the two ends, and this results in the circularization of the genome. But this circularization has been related or that is involved in replication, but not in translation. The translation of this virus, the flavivirus, is, or they use a mechanism which is a cap-dependent mechanism, which is somehow similar or is the consensus mechanism that uses the messenger RNAs in eukaryotic cells. But we know that the 3' UTR of the West Nile virus enhances translation of the virus. So you can see here the deletion of the 3' UTR results in a reduction in translation. So the question, and besides that, we demonstrated that it exists a cap-independent translation mechanism, which is also controlled by the 3' UTR. In this case, it's absolutely dependent on the presence of the 3' UTR. And this effect can be achieved by the addition of the 3' UTR in trans. And as I have mentioned, it was shown that there are complementary sequences between the two ends, and these two RNA fragments can come together and form a complex. But as I said, this complex seems to be related with replication and not with translation. So the question is, which information contains this 3' UTR domain or the different structural units to achieve this functioning of regulating the translation efficiency. So in order to understand that, the first thing we tried was we wanted to see whether the 3' UTR is able to recruit or to bind the ribosome. So we did a single experiment in vitro, again, just with the RNA to test whether the RNA was able to recruit a purified 40S ribosomal subunit. And as you can see in this figure, it occurs, and as a preliminary data, our preliminary data indicates that there is more than one site, or it seems to be more than one site of binding or recruitment of the ribosome within the 3' UTR of the virus. This interaction is specific, can be competed by the same unlabeled RNA, but is not competed by an unrelated RNA. Further, we demonstrated that this interaction depends on the ribosomal protein, so it's an RNA-protein interaction. Further, to continue with this characterization, we wanted to determine the nucleotides or structural units that are important for this binding. So we did again this structural analysis, and by comparing the structure in the presence or absence of the 40S subunit, ribosomal subunit, and we identified two areas that seems to accumulate most of the changes. That this is the area that includes this structural unit named SL3, and this other area that includes two structural units that they are known as dumbbell, the 5' dumbbell and the 3' dumbbell. So these two areas postulate as the potential binding sites for ribosome. In a new experiment, we try to analyze or to determine the nucleotides that are important for the binding, because they could be directly involved in this binding, or they could be involved in the formation of the structure that is competent for the recruitment of the ribosome. And once again, most of the nucleotides are in this area, including the SL3, and in this double dumbbell area that I described before. And finally, we did experiment, a toy printing assay, which is the classical assay in which, after binding the ribosome using specific primers, we do a primer extension and determine the points in which the reverse transcriptase stops, which means that that because it hit with the ribosome, so it gives information of where of the binding sites. And once again, as you can see, we determined that the SL3 and the 5' DB seems to be potential binding sites of the ribosome. But interestingly, the 5' DB is involved in the formation of this interaction to form a pseudonode that is named PK3. Pseudonodes are a higher order structural motifs that they are functional and have been involved in different functions, but they are characterized, or one of these functions is to recruit specific proteins or specific factors, even they have been related with the binding of ribosomes. So we wanted to see whether this pseudonode is involved in the binding of the ribosome. So we made different constructs to avoid the formation of this pseudonode structure by deleting the 3' or the 5' of this, let's say, the sequence involved in the formation of the pseudonode, or we use just a construct in which we just have the domains of the sequences necessary for the formation of the PK3. And as you can see in this figure, only the wild type and this fragment, so those constructs that support the formation of the pseudonode can bind or recruit the 40S ribosomal subunit. whereas constructs where the formation of the pseudonode is not possible cannot bind the ribosome. So now we wanted to see whether these interactions that occurs in vitro in the absence of any other factor, just with the RNA, could be functional. So using a construct similar like the ones I described it before, so the luciferase messenger flanked by the 5' UTR and 3' UTR of the West Nile virus, we tested the translation, and we made different deletions. We deleted the 5' DV, the 3' DV, the pseudonode, and as you can see here, these three constructs result in a reduction of translation. This is the control. And similarly to the complete deletion of the 3' or the 3' UTR. But interestingly, when we deleted the other potential binding site, the SL3, it results in an increase in translation. So almost 50%. This resembles the result I showed you before that we obtained with hepatitis C virus. So it means that this element, structural unit, seems to play a negative role or a negative regulation role of translation. So basically, what with this experiment, we can conclude that the 3' UTR of West Nile virus seems to use the recruitment of the ribosome at the 3' UTR as a mechanism for regulation of translation. It's also interesting to see that we include in this study a deletion of this last structural unit, the 3' SL, which is this one. And as you can see, the deletion of this structural unit has the same effect than the complete deletion of the 3' UTR. But we didn't detect any with our structural study, we didn't detect that this site could be involved in the recruitment of the ribosome. But it's important to say that the studies that we did, the structural studies, were done only with the 3' UTR. But this functional study was done in the presence also of the 5' UTR. So we wanted to see now whether the presence of the 5' UTR modifies the structure at the 3' UTR. So those are the toy printing assays that we obtained just with the 3' UTR. But when we add the 5' UTR, you can see that first we maintain the SL3 as a potential binding site, but the 5' DV, let's say the stops in the 5' DV becomes weaker, and this move or appears a new potential binding site at the 3' SL. So it seems like the presence of the 5' UTR modified this structure, or somehow, let's say, helps to reposition the ribosomes at the 5' end of the genome, so at the bottom of this structural unit. So it seems to be also a communication between the two ends of the genome, also in translation. Similarly, we did the same experiment to see what happened at the 5' UTR. So using these toy printing assays in the absence of the 3' UTR, we detect the stops at this position, which determines that the binding site is this area, which is expected because here is the star codon. But when we add the 3' UTR offset, okay, the 3' UTR, we could see that there is a stabilization of this interaction of the ribosome at the 5' UTR, and somehow a repositioning of this, and we detect this stops at the 3' of this sequence. So once again, as we detected for the hepatitis C virus, it seems like the two ends of the genome, the 5' and 3' UTR, comes together and modulate the structure and somehow helps to reposition the ribosome. Finally, we tested whether this is a general mechanism of regulation of translation in all flaviviruses. For this purpose, we test the ability of the 3' UTR of other flaviviruses, in that case dengue virus and yellow fever virus, to recruit also the 40S subunit. And as you can see in this figure, both of them are able to recruit the 40S subunit in a more efficient way than the West Nile virus, which is this one. And as you can see in this figure, the three of them contains this 3' SL structural element, and also they contain at least one of these double structural units that we postulate that could be the potential binding site of the ribosome in these other flaviviruses. And interestingly, none of them contains this SL3 binding structural unit that we determined as another potential binding site and which has a negative role in translation. So finally, we made a set of constructs in which we are using the luciferase gene flanked by the 5' UTR of West Nile virus with cap or without cap, and at the 3' UTR, we used as a control the 3' UTR of West Nile virus, and we replaced that, or we replaced it with the 3' UTR of dengue or yellow fever. And as you can see in these figures, both in mammalian cells and in mosquito cells, the presence of the dengue 3' UTR or the yellow fever 3' UTR result in an enhancement of translation much higher than the one that produced the West Nile virus. This is very interesting because it's telling us that there is a correlation between recruitment of the 40S ribosomal subunit at the 3' UTR with the enhancement of translation. And interestingly, in these two 3' UTRs does not exist or doesn't exist the SL3 element, which has a negative role in translation. So finally, as a take-home message, I would like to say that viral RNA genomes possess an information storage system that overlaps with and complements the protein coding system. They store essential information in structural RNA units. Viral RNA genomes are a succession of cis-acting non-coding RNAs which play important functions without needing to be translated. And so the interfering with their structure and therefore with their function may constitute a promising antiviral strategy. And here I want to acknowledge the people of my lab, Dr. Cristina Romero Lopez, who is running the lab together with me. She is responsible for all the work that I presented done with hepatitis C virus, and also Sarah and Beatriz that have been involved in the work, mainly in the work with the West Nile virus. And this is all. And I'd like to thank you for your attention, and I'll be glad to answer any question if you have. Thank you very much.